Alrighty, so welcome back to B2.1, Membrane and Membrane Transport. We were finishing up talking about passive transport methods with osmosis. Quick side note, because I just opened this, Mr. Harenko, aka Colin, stopped by my room at the end of the day to deliver me my birthday Christmas gift. My birthday was last month and Christmas was 2 months ago, but he gave me custom stickers of my dog. So guess what everyone's getting when they win Book-It for like the next 2 weeks? I'm so excited. Why he picked this god-awful image, I don't know, but it's fabulous. And I'm so hyped. Anyway, that aside, he also gave me a card with a bird on it. I have never really liked birds, but that's okay. It was worth it. I'm going to tape it to my wall. Alright, so osmosis. Where are all the highlighters? The pictures of my dog really got me caught off guard. And I could not wait. People who watch this before tomorrow morning needed to know before tomorrow morning. So osmosis is essentially just diffusion but with water. Like, to really make it as easy as possible. Osmosis, what color was diffusion? Purple? I think it was purple. So osmosis is diffusion of water. Specifically from an area of low concentration of solute, aka high concentration of water, to an area of high concentration of solute, aka like low concentration of water. And so like if you're looking at different solutions, there's a bunch of special like terminology for water related solutions because they just like to make things difficult. And so something that has a low amount of water, high amount of solute is hypertonic. Oh, where am I going to put this? I guess I'll put this down here. Something that has a high amount of water, a low amount of solute, is hypotonic. I like to think hypo kind of sounds like low. I don't know. And like the big rule of thumb to help you remember which way it would go, is solutes suck. Number one, because their questions are always obnoxiously worded, but really it's because they suck water to them. So something with a lot of solutes is going to draw the water towards it. And so if we look down here, we have, no, it's fine. So we have our hypertonic solution, hypertonic, with our solutes. I was going to make them a different color, but I don't know where my red highlighter is. It's not where it needs to be. So these sucrose molecules are just going to be colored in pen. It's going to have to be underlined. That's so unfortunate. Is your high solute concentration, hypotonic. Has your low solute concentration, or in this case, none. And so your net movement of water through osmosis is going to be this way. Do I color all the waters? I really want to. No one can stop me. Okay, my heart's happy now. So osmosis is due to the difference in concentration of substances dissolved in water, aka your solutes. And so when they're dissolved, they become surrounded by your water molecules because of the hydrogen bonds, and it restricts the movement of the water, which makes that area a higher concentration of solute. It has fewer water molecules available to move, which then creates your concentration gradient. And so in this one, your net flow is this way, because all these like little ones, I'm not going to color in the solutes in this one. With the pen, that would just be too obnoxious. But you have your waters. And although it looks like there's a fairly even amount because so many of them are bound, it creates your concentration gradient. These ones are loosey-gooses, the other ones are bound. These ones are less satisfying to color. There we go. And so what molecule what molecule diffuses across the partially permeable membrane during osmosis? That's water. Write it in giant all caps letters, because one thing IB really likes to do, I've been looking through the question bank because I've been putting together a little quiz. IB really likes to throw you trick questions wherein it's not necessarily that you need to know what's happening on the graph or that you need to know exactly what they're on about. It's that you need to know what osmosis is. And so it'll be like chlorine ions like go across them or are at a gradient and they're in solute. And what's moving to cause the change in concentration? And the answer is water, because it's osmosis. The chlorine is not going to move unless there's some sort of like pump or channel. The water is going to be what diffuses, and so don't ever say it's the ion. If you have an option and the question's about osmosis or water moving, it's always the water. If it's about osmosis, it's always the water that moves. It's never the ion. Unless it specifically states that the ion's moving through like a pump or a channel, it's always the water. Pro tip. And so what's a solute? It is just something dissolved in water, or a solvent. Oh, I'm not going to write that in purple. That's the diffusion color. Oh, what's a solute color? I guess it's red now. Okay. A particle dissolved in a solvent, usually water. For the purposes of IB Bio. My favorite example that I used to give to all the middle schoolers is like Kool-Aid powder. Is the solute in water. That's the solvent. And then Kool-Aid drink. That's the solution. I don't know why that's the example I came up with, but it works. They remembered it. I hope. Maybe. Who's to say? And so, what is happening? Normally, hydrophilic and polar molecules don't diffuse easily through the membrane. Why does water, which itself is polar, move without difficulties through the membranes? It's small. It's small and it's not charged. It's small enough that it can sneak its little way through there. Some cells have aquaporins. What are they? They're just channel proteins for water. They increase permeability of water greatly. They allow water to pass through much easier. And so tissues, oh, you can't even see that question. Scrooge. Tissues where you see a lot of aquaporins are things that take in a lot of water, so like your kidneys, like kidney cells, I guess. And in plants, root hair cells. Things that need to take in water. That's pretty much it. Oh, here's your aquaporin. Come on. Let's outline her. Oh, I should have just highlighted her. Oh well, it's too late now. I'm already doing this nonsense. Where do I draw this arrow? There we go. Aquaporin. See the water going in? Look at her go. I don't know, man. I'm having too much fun. All right, flip. Please focus. Please. 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 Whoa. Okay. Alrighty. So now we have to look at active transport. Your key example that you should recall is your salty banana. You see the salty banana here? Look at it go. Here's the banana. Give me a second. It'll look more like a banana, I promise you. Hang on. I have enough colors for this. It's got your K plus. And then, what color's a salt shaker? I guess gray. An A plus. That almost looks like a peppery banana, but it's salty, I promise. Okay. So, ooh, I forgot what color we made active transport. I think it was supposed to be red, but then I goofed it up, and I think it's actually pink. Yeah, protein pumps are pink for no real reason. Where's the pink one? There we go. So, active transport and protein pumps. And so not everything diffuses easily, specifically things that are hydrophilic and charged. They can't go through. They need to have either a channel or some type of active transport, depending on how large they are. If they are large and or in charge. And so active transport of substances through protein pumps. They're transported usually against your concentration gradient. Oh, come on. Protein pumps transport things in only one direction. They need energy to have some sort of conformational change or like a shape change. And the mechanism requires energy. Why is that written here twice? Am I stupid? I don't know. Oh, one of them requires energy, one of them uses or releases energy to have the shape change. Okay, whatever. Good enough. Alrighty, so we have... Oh, let's annotate our little picture while we're at it. Where's the pink pen? So we have... requires energy. Shape change. Travels in one direction. And how am I going to label this? Against gradient. So it would go this way and that way. That's the dumbest way I could have made that look. Oh well, it's fine. It's too late now. Alrighty, so now let's compare active transport using protein pumps to facilitated diffusion, which I remember was green because I remember I was like, wow, Jessica, how clever. he made it green, and proteins were also green. And it was entirely an accident. And what color are we going to put on top? Pink and green make like doo-doo brown. I guess that's what we got then. So they both use integral membrane proteins. They both transport substances across the membrane. They're like dumb as white. They both transport substances that cannot pass on their own through the membrane. And that's really where the similarities end. For active transport, it goes in one direction. One direction. You are beautiful. Requires energy. ATP. And it moves against concentration gradient. Low to high. Facilitated diffusion can go in two directions. No energy required. And it moves with the concentration gradient. Up from high to low. Bump it up. And then you have some different types of protein pumps. You have uniporters, which can move one specific ion or molecule, uni, one. You have a symporter that can move two different molecules, both in the same direction. I like to think like symbiosis, like they're going together. Then an antiporter, which carries two different ions or molecules, but in different directions. That's like your salty banana. Okay. On to the next one. Good to look at the fatty acids again. So your fatty acid composition and like the specific fatty acids and the types of fatty acids that you have, like the variations between them, actually has a lot to do with how your membrane functions. And so they're the main structural component. You have You have your phosphate group. Which replaces one of the typical 3 fatty acids on a normal lipid, and it makes your polar head and your 2 nonpolar tails. And so your amount of saturated and unsaturated fatty acids in your phospholipid bilayer is regulated such your membrane remains fluid but strong enough to avoid becoming perforated or having gaps. And the ideal ratio depends on the temperature. So an Arctic fish would have a higher percentage of unsaturated fatty acids in their membrane than fish from warmer waters or temperatures or climates. And so it really just depends on like where that organism lives. And so let's mark off our fatty acids. They are our nonpolar region. Oh, I should have done this in red, not pink. Oh well, too late. I don't know where the red one is, honestly. Fatty acids. Oof, that was nasty. I did not do a good job there. Oh, I have so many more. It's okay. We'll do this later. And then our polar head. Ooh, can we do it in one swift motion? No, we can't. Elegant. And then emphasis on saturated. There are straight ones. Are unsaturated is the bent. Bump, ba-da-da, scooch. Bump, ba-da-dump. And so how does the ratio of saturated to unsaturated fatty acids affect membrane fluidity? And then how does the, come here, length of fatty acid tails affect membrane fluidity? Those look like the same color. I promise you they're different. So first, we'll look at saturated to unsaturated. So more unsaturated fatty acids means it's more fluid. It means they're more spaced out. They have more wiggle room. And less double bonds. Makes it more viscous. It's like this would be like if we had like more saturated. And then number 2, shorter tails, makes them more fluid, lower freezing or melting point, scooch. Membrane less likely to freeze. And so here's what I mean by like the more wiggle room. When they're more unsaturated, they have like literally more space between them because they're bent, they're pushing each other away. When they're more unsaturated, they have more wiggle room. But when they're more saturated, they have less wiggle room. They're able to be packed more densely. They're more viscous. You have less of those double bonds because those double bonds are what creates the kink. Less double bonds. More wiggle room. And so your ideal composition depends on organism temp. Not too viscous. Not too rigid. Not too fluid. Oh shoot, you can't see that. Sorry. Like, you don't want it to be mush, but you also don't want it to be like solid as a rock. Does that make sense? You want it to be fluid, but not like mashed potato goo. And you want it to be sturdy, but not like cell wall kind of sturdy. Speaking of making sure we have a balance of fluidity, it's time to talk about cholesterol. Get out of here. Please, 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 please. Alrighty. Cholesterol in my brain is yellow, so we're just gonna make her yellow. Oh, come on. Cholesterol and the membrane fluidity in animals. So cholesterol is important. It helps with stability and fluidity of membranes at temperatures outside of optimum, so super low or super high. Otherwise, your membrane becomes kind of compromised. And so what chemical properties do the steroid ring and hydrocarbon tail have? So this part, they are, oh, where's the red? Get lost. They are hydrophobic and nonpolar. What chemical properties do your fatty acids of your phospholipid have? Crazy, they're hydrophobic and nonpolar. Don't ask me why there's a capital L in there. What chemical properties do your hydroxyl OH head have? Hose love the water. They're hydrophilic. And polar. What properties does the phosphate group of your phospholipid have? It's hydrophilic. And polar. So essentially what they're getting at is they're twinsies. They have really similar properties in terms of like how they're structured, which allows them to do this like kind of crazy like little link up thing. So what your cholesterol essentially does is its head binds to the head of one phospholipid, and then its like weird tail thing binds to the tail of one of like an adjacent phospholipid. And so it helps to kind of keep your membrane like glued together. It's like an additional little like structural component here that helps it to stay more stable. So yeah, pretty cool. They have like mirrored properties, which means I know you know what time it is. It's time to decorate. And so we'll give our bestie the boots. They're gonna be kind of weak boots because he's got weird legs. They gave this one janky legs. And he really loves the water. And his friend also loves the water. Oh, they're both hosed together. They're gonna be like sharing a drink like little, like lovers. Here's their cup. For their water. H2O. He doesn't really have like a feet like that, so we're just gonna give him like a rain poncho. There's his poncho. It's a poncho. And then of course gotta stick with our color coding. Hydrophilic gets blue. Why'd they have to make them all chemical structure like this? Make them like a little staircase. I still don't know where red is. I'm like really bothered by that. He's calling janky legs. Beautiful angels. You can do them down here too. Scooch. Scooch. Oh, I need the other side. That's not going to go well for me. Our hydrophobic region. Oh, I did not color that very well. Oh well. It's too late now. And then our hydrophilic region. It's colored like doo-doo. Okay, it's fine. Polar head. Water loving hoe. Non-polar rings. And hydrocarbon tail. Non-polar rings. Hydrocarbon tail. Oh, they have it here too. Non-polar tails. Non-polar rings. Non-polar tail. Hydrophilic head. Hydrophilic head. I crack myself up. Hydrophilic heads. Oh, and this time, let's highlight all of our cholesterols in yellow. Boop. Get a little change of pace going. I feel like they're way too tiny here for me to try to color code them, so. Well, now the fatty acids also need to be colored, otherwise it's gonna look weird. And then they wonder why this takes me so long. You know, I counted it. I was like, oh, it's only 7 pages, Jessica. This should not take that long. I don't know who I was lying to. All right, now time to do the write-up. So, your OH group, or your HO group, oh, shoot. Okay. Of cholesterol. Gosh, okay. Links to phosphate head of phospholipid and it's like Wolverine. Steroid, hydrocarbon, tail of cholesterol, joins to the fatty acid tail of a neighboring phospholipid adds additional like glue and binding to help maintain fluidity and stability. More on how momentarily. Oh, please. Not you. Close all these, oh my god. Like every pen and its mom open. Scooch, scooch, scooch. And so without cholesterol, submarines would be too fluid, not firm enough, too permeable, turn to mush, turn to grossness. Trash. Alrighty, so now let's look at temperature and why this is so important. Bum-ba-da-dum. Ooh, okay, so let's make without cholesterol, like brown, doo-doo brown. So this one, without cholesterol, is this doo-doo. No, the lights are turning off. There we go. With cholesterol, this is like cold, and this is like hot. This is like a solid, this is very like fluid mush mush. Oh, shoot. Low temperature, no cholesterol. High temperature, no cholesterol. It's low temperature, so we'll make it blue. It's high temperature, so we'll make it as close to red as I have currently. RIP my red one, I really don't know where it went. And then with cholesterol, at any of the temperatures, she's a beautiful, gorgeous, she's golden. We love her. Scooch, scooch, scooch. And so what cholesterol essentially does is it acts like a little buffer. Like it kind of helps to contain what's happening. And so at low temperatures, things get to be too rigid. At high temperatures, things get to be too fluid. However, with cholesterol, things tend to stay very stable because at high temperatures, it stops a lot of that excess movement of your membrane particles. And at low temperatures, it provides a little helpful gap to keep it from getting too like clumped together. So it's like a win-win. It's fantastic. And so first let's talk about, oh gosh, get out of here. What happens without cholesterol? I guess we'll write that in Dookie Brown. And so, without cholesterol. So at low temps, tightly packed and very rigid. At high temps, very mobile and fluid, mush. So now let's look at with cholesterol. I'm writing it in yellow, I'm doing it. Can't stop me. So low temps, what is happening? Cholesterol disrupts the aggressively tight packing caused by cool temps and fluidity is increased. Prevents it from becoming just a brick. High temps, cholesterol contains, I guess I should have said constrains. Oh well. Constrains the excess loosey-goosey movement and decreases fluidity to maintain form. Overall, it acts, acts as a stabilizer and a buffer. Boop. And that's that on cholesterol. So now we're going to look at vesicle movement. Oh, this is the one that's red. I really need to find the red one. It's really going to drive me insane. Let's see. Okay. I found it. I don't know why it was in the drawer. So membrane fluidity and the fusion/formation, of vesicles. And so they gave you like a little chart. I think this is kind of hard to look at, if we're being completely honest. So basically your fluidity of your membrane allows it to change shape, to break and reform. During endo and exocytosis, ATP is required for this process to happen. So, key things, ATP is required, and your bilayer is always connected. When you look at this like little picture here, so what you're looking at is like a zoom in of this like little section here. So this side's your cell, this side's your vesicle. You can see when they meet, you can see they form one layer. And then this isn't them like breaking. If you notice, it's like this like butt cheek thing where they both kind of fold outwards. Like if you were to draw in the other side, it would be like that. So they're never being broken. This is just what it looks like when you zoom all the way in on this little section. So it's like a very zoomed in view of right here. And so your vesicle formation and exiting, I feel like, is pretty straightforward. Vesicles either just kind of arrive at the membrane, join, release, or they like form around something, butt off in, and then move to wherever they need to be in the cell. Like, it's not super duper complicated. You have your 3 different types. You have, phagocytosis, pinocytosis, and receptor-mediated endocytosis. You don't need to know a ton of detail about each type, but like just knowing the basics is pretty important. So phagocytosis is taking in large particles solids. So bacteria, organic matter, like food. Penocytosis is taking in liquid. Ooh, what is happening? Taking in liquid. This is apparently a liquid. Image is awful. And then receptor mediated. This is the only one that's kind of weird. So there's like a receptor coated pit. There's a coated pit with receptors. When enough ligands bind, two receptors, coated pit breaks off, and takes in the ligands. Like, this one's weird, okay? You really won't see receptor mediated super often. Here's your little coated pit. You have your ligands binding. When enough of them bind to the receptors, it just kind of does the little vesicle thing. That's it. Okay. So now looking at endocytosis versus exocytosis. I think, what's my smartest plan of attack? Where's the green? We'll make endocytosis green for enter. Get out of here. Dun, 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 dun. Examples. Oh, you guys can't see this bottom part. Okay. Will you please scooch? Scooch. It gives you examples down here too. And then we'll make exocytosis red because it's exiting. That means the differences are going to have to be, or the similarities are going to be brown again. That's so unfortunate. Oh well. And I don't like that this doesn't line up. That makes me very sad. It's fine. Alrighty. So key things. Endocytosis, I like to think. Enter. It goes from out to in. Exocytosis, I like to think. Exit. It goes from in to out. So differences occurs by invagination, which is just infolding. You know how like a vagina is folded? Invagination is just infolding of membrane. Forms a vesicle. And detaches into cytoplasm. Your examples: transport of like large hydrophilic particles, so like food in single-celled organisms, Fetuses uptaking antibodies. In the womb. Phagocytosis of bacteria by phagocytes. And uptake of viruses by white blood cells. Get lost. For exocytosis, it occurs by a vesicle made in the Golgi fusing to the membrane and releasing contents to the extracellular space. No receptor mediated, which makes sense, like you wouldn't necessarily have receptors like that on the inside. Examples, protein synthesis, in the rough endoplasmic reticulum with vesicles moving polypeptides to the Golgi, secretion of enzymes from pancreas cells to pancreatic duct, and then to intestine. Release of neurotransmitters, from synapses, or like acetylcholine, in neurons, and then sweat and breast milk secretion. Come on. For similarities, membrane proteins facilitate both processes. Using ATP. For large particles. Require vesicle transport inside cell. I almost wrote inside side. Inside cell. Need ATP. Alrighty. Zoom out. So these next couple pages go over gated ion channels and neurons, which I'm not going to spend a ton of time going over with you because we went over them we're going over them so aggressively in class with your salty banana packet. So I feel like you can review these if you need to. Same with this page like the second or third, page 10. Fill this out, use the reading, use your salty banana packet, do this one on your own. I would rather spend time like going over the parts that we didn't go over a ton in class. And so one example of something that we didn't go over a ton in class is another example of Co-transport and active transport that uses a very similar sodium-potassium pump to the one used in neurons. What color should we make this? What color haven't we used? I feel like we have not used blue a lot today. I know, you know what? We've really been slacking on purple. We'll make this one purple. So sodium-dependent glucose co-transporters as an example of indirect active transport. And so in your small intestine, apart from its function of enzymatically breaking down macromolecules into monomers, your digestive tract is primarily responsible for the absorption of food and nutrients. Your absorption of food takes place in your small intestine, where your villi, which are your folds in your intestine, and your microvilli that help enlarge your surface area. Your glucose cannot pass through your plasma membrane by simple diffusion because it's polar and hydrophilic. Therefore, a co-transport system with sodium ions is put in place. And so here's like your steps broken down of how it happens. The next couple questions we're going to look at also kind of break it down. And so here's essentially what happens. You have, where's the purple pen? There we go. You have your glucose and your maltose. Your maltose is hydrolyzed to become glucose. Your oh my gosh, I had such a brain fart. I'm so sorry. Your glucose is co-transported with your sodium down its concentration gradient, and then you have a pump to help increase the amount of sodium outside. That's really it. And so why can glucose not get into the villus of the cell, or of the small intestinal cell, by simple diffusion? Because it is polar and hydrophilic. And so what type of transport, active, passive, simple, facilitated, gets glucose into the villus of your small intestine? It is coupled co-transport with Na plus via facilitated diffusion. What type of transport pumps Na ions out of the villus? Active. What is the purpose of your sodium-potassium pump in this case? To increase amount of sodium ions outside the cell to allow facilitated co-diffusion of glucose with sodium ions back in. What drives the co-transport of glucose into the villus? The concentration gradient of sodium ions. Okay. Pretty simple. I feel like once you, like, now that you understand, like, how, like, the axons work, I feel like this is pretty easy. Sodium's being pumped out to make sure it has high concentration outside, so that your glucose and your sodium can then facilitate a diffusion in together. That's really it. Bump it up. Fun fact, sodium and glucose do this all over the place. Because here's sodium-dependent glucose cotransport in your kidneys. So in your kidneys, the same system of inactive or secondary transport is in place. Your kidneys are a whole mess. And so your sodium outside your tubules is increased through active transport, allowing a concentration gradient to build up, which then allows your glucose to go back into the cell to filter it out of your urine. So kidney's initial filtrate is nearly 5.5 kilograms of glucose. Urine does not contain anywhere near that much. It's reabsorbed back into the blood. And this is how. So, I know this has numbers on it. Ignore those numbers. I'm going to write my own. So here's step 1. And here's step 2. I don't like that they don't draw the protein channels in, but that's okay. So step 1, your sodium, potassium, pumps, move sodium out of kidney cell via active transport to create concentration gradient and then step 2, sodium and glucose co-enter the cell through facilitated diffusion due to the concentration gradient of sodium ions. So same thing. And then your last thing is your adhesion of cells to form tissues. Main thing you need to know is that your peripheral and integral proteins have a lot to do with cell-to-cell joining. There's a lot of different types. You can have interlocking junction proteins. You can have proteins that form like an intercellular space. You can have channels between them. They're different CAMs, or like cell adhesion molecules, and they just have different joints depending on the function. That's really all that you need to know. They're just a protein that's embedded in your phospholipid bilayer that project outwards, and they bind in different ways to form tissues. And that concludes everything you need to know. Oh, for your last page, just as like a test of how well you know it, I do want you all to fill this one out. I want you all to fill it out on your own using what you learned. Go through, complete your final page. P or A for passive or active. What does it involve? A concentration gradient. What moves through it? Does it involve any other pieces like aquaporins, proteins, vesicles, protein pumps? And then like at least one example for each one. And that'll be part that I give you feedback on before I pass this back. And you should be good. Good luck.